Hello friends, uh, today we are going to discuss IIT Jam Biotechnology 2015 question paper. In this video we will be discussing section A and B, and in the upcoming videos we will be discussing the section C. Okay, so first question. Which one of the following most accurately describes the process of natural selection? So we have to choose the option which is accurately describing the natural selection. Selection of one species over the competing species. Selection of individuals that successfully defend themselves against the enemies. Selection of individuals that produce more than the average number of offspring. Selection of individuals that are more attractive to the opposite sex. See, what does natural selection say? Natural selection says that survival of the fittest. So, which species is going to be more fittest? Which is going to produce the more number of offsprings. So, who is going to produce more number of offsprings? Who can attract more number of uh, its opposite sex? And next is they can save themselves from the enemies, and then also producing large number of offspring, so that if any of the destruction of the species, the other remaining population can survive. Suppose there are 100 members, okay, from that 100 members, suppose 40 members died. So from that, at least there will be 60 members who can further reproduce and double this to 120, right? So it is important that we have a higher number of um offspring of that species. So selection of one species over a competing species, this is right. This describes a natural selection, that means survival of the fittest, which can compete better, can survive better, right? Selection of individuals that successfully defend themselves against enemies, right? This is also correct. This is also survival of the fittest. Selection of individuals that produce more than average number of offsprings, yes, this is also true. And also selection of individuals that are more attractive to the opposite sex. So see, if they are more attractive, they are going to produce more number of offsprings. So this option is also the correct option. Okay, now coming to the next question. Identify the statement that is true for of operons. Okay, so first see the first point. Let's see. Fine regulation of expression of individual genes are made possible by operons. So individual genes is given here. But this is not the case with operons. What happens in operons? Suppose here is a promoter, here is an operator. So they are consecutive genes present here, one after other. This is operon. Okay. If it is an individual gene, this is a case of eukaryotes then. Whereas operon is in the prokaryotes. So this option is wrong option. They are asking the true option. Only genes involved in the carbohydrate metabolism are present in the operons. This is absolutely wrong, because operon also operates the metabolism of tryptophan. Okay, we know that. So this is a wrong option. Tryptophan is what? Amino acid. Next, feedback inhibition of biosynthesis of multiple enzymes by a single small molecule is made possible by operon. This is the correct option. Okay, let's see the next. In the case of inducible operons, the inducer binds to the operator. Okay, inducible operon, the inducer binds to the operator. No. Here, what happens in the operator, when the repressor is present, when it has to induce the expression of genes, the inducer is going to bind on the repressor. Okay, not on the operator. So this is the wrong option. Okay, coming to the next question. Signaling pathways usually comprise of several intermediate steps that are arranged in form of a cascade. What is the primary outcome of such arrangement? What arrangement they are talking about? In the signaling pathway, what happens when a receptor is bound to the ligand? This will, start a signaling pathway. This is going to, signal to the first, then this to the second, third molecule, fourth molecule. By this signal is increased to about 1,000-fold. Okay, so what is happening here? The amplification of signal is taking place. So let's see the option. Signal specificity of signal transduction. See, they're not asking that. They're asking that what is happening through the signaling cascade. Okay, this is a wrong option. Specificity of the cellular response? No. Amplification of the cellular response? Yes, this is a correct option. Fine tuning of the cellular response? No, this is a wrong option. So correct option is C. Coming to the next question. Which among the following contains oxygen-rich blood in the human vascular system? Right ventricle? No. Aorta and pulmonary vein contains the oxygen-rich blood. So 2 and 3 are the correct options. So D is the correct option here. See, aorta will contain the oxygen-rich blood. It is going to supply to all the organs. Okay. Pulmonary vein is a connection between heart and lungs. Okay, the lung is going to send the oxygenated blood from lungs to heart. Okay, so 2 and 3 are the correct options here. Coming to the next question. Choose the option that lists the correct sequence of steps involved in the gene therapy. So what is gene therapy? Dekho, therapy is what? You are correcting something is default and you are correcting that. In the gene level, when we are doing such a thing, we are calling it as gene therapy. Okay, so what happens in gene therapy? Injection of expression vector into the patient. Yes, we do this. Why expression vector? Suppose the patient is lacking some protein. So what we do? We inject the vector into the patient and that vector is going to express the proteins, and that proteins express that genes, and that genes are going to produce RNA, and from RNA it is going to produce proteins. So this is the correct option. Wild type genes is inserted into the expression vector. Yes, so they are going to do what? They are going to insert the wild type normal gene into the vector and insert into the patient, so that this patient, if it is defective in that gene, it is going to express the correct gene, so that it can produce a protein. Right, now this patient is not producing that protein, but now this is producing that protein. So this is a correct option. Wild type gene is isolated and cloned. Yes, this is correct. Wild type gene is transcribed and translated in the patient. Yes. So all the options are correct. P, Q, R, S. So this is P. No, there is no R in this. So D is the correct option. P, Q, R and S. Yes. Okay, coming to the next question. Cephalin, a biological surfactant, is choline phosphoglyceride? No, this is what? This is ceramide. Okay. Ethanol amine phosphoglyceride? Yes, this is the correct option. Glycosphingolipid? No. Sphingolipid? No. Okay, coming to the next question. Which part of the genomic DNA contains the sequence corresponding to the 5' untranslated region? So this exon, intron, upstream of the transcription start site and upstream of the promoter. So here the right option is exon. Exon contains the 5' untranslated region. So we have 5' UTR and we also have 3 prime UTR. Okay, so this comes under the exon. Okay friends, coming to the next question. Which one of the following is not true of RNA polymerase 2? What is not correct? Okay, it requires a primer to initiate the transcription. What is RNA polymerase going to do? It is going to produce RNA. Okay, RNA polymerase 2 is going to produce the RNA. But we know primer is required for the DNA replication. It is not required for the production of RNA. So this is a wrong option. It makes an RNA copy of one strand of double-stranded DNA at any given time. This is correct. Okay, so not true they have asked. So this is a correct option here. Okay, this is a correct option. It makes RNA copy. Yes, this makes. So this is a wrong option. It does not synthesize rRNA and tRNA. Yes, it does not synthesize. Okay, it catalyzes the, who produces rRNA? It is produced by RNA polymerase 1. Okay, tRNA is produced by RNA polymerase 3. Whereas RNA polymerase 2 produces mRNA. Okay, it catalyzes the polymerization of RNA only in 5' to 3' direction. This is also correct. This is wrong here. Okay, next question. Choose the option that shows the correct pairing of the cellular components with their corresponding function. Okay, friends, let's discuss this. So, dynein, what is its function? It is going to form the membrane vesicle. Okay. No, sorry friends, it is not going to form membrane vesicle. It is going to help in the beating of the flagella. Okay, this is response for the beating of flagella. Okay, kinesin, we know this very well. Kinesin is responsible for transporting the organelles. Okay, so kinesin is going to be 1. So let's see where S is 1. Okay, S is 1 in the B option. So it is for the movement of organelles. Endosome is for the membrane vesicle. This is a membrane vesicle. Desmosome is going to do what? It is for the attachment of cell together. We know this very well, that two cells are joined together by desmosome. Desmosome. Okay, so B is the correct option here. Okay, an enzyme shows highest activity in the pH range of 2 to 3. Okay, this is the acidic medium. Okay, at pH 4 and pH 7, the enzyme exhibits 50 and 1% respectively of the highest activity. Which one of the following states of an amino acid residue in the catalytic state is most responsible for the activity profile? Okay, so here the correct option is going to be a protonated asparagine. Okay. Oh, sorry, aspartic acid. Okay, aspartic acid. Protonated form of aspartic acid. Next question. The specific productivity of cellulase production by Aspergillus niger follows a linear relationship with the specific growth rate, mu, and is of form QP, alpha, mu plus beta, where alpha and beta are constants. Assuming that the value of alpha and beta are constant respectively, which one of the product formation kinetics is true? Okay, so for this the correct option is non-growth dependent kinetics. Okay friends, now coming to the next question. Choose the option that shows the correct pairing of the diseases with their corresponding causative organisms. So, see, malaria we know is caused by Plasmodium falciparum. Okay, plague also we know it is caused by Yersinia pestis. Sleeping sickness is caused by Trypanosoma gambians. So the left option is Chagas disease is caused by Trypanosoma cruzi. So the correct option is B here. Okay, coming to the next question. Choose the option that shows the correct pairing of the products with their corresponding microorganisms. Okay, so citric acid is responsible for the production of Aspergillus niger. So Aspergillus niger will produce the citric acid. Okay, and who produces polyhydroxyalkonates? Ralstonia eutropha is responsible for the production of polyhydroxyalkonates. Ralstonia eutropha. Ethanol is produced by Zymonas mobilis. These things we have to remember. Okay, these things are to be learnt. So C is the correct option here. Coming to the next question. Determine the correctness or otherwise of following assertion A and reason R. Assertion. B cells secrete antibodies against virus while cytotoxic T cells kill virus infected cells. Yes, this is absolutely true. B cells, they produce antibodies against virus while cytotoxic T cells, they directly kill the virus infected cells. Okay. Reason. B cells confer active immunity while cytotoxic T cells confer passive immunity. No, this also confers the active immunity. So R is false. So A and R are true, and no, this is wrong. A and R are true. No. A is true but R is false. This is the correct option. C is the correct option here. Okay, next question. Which one of the following options shows the correct pairing of enzyme with its corresponding application? Okay, so papain. Papain is a meat tenderizer. Okay, bromelain is responsible for gluten complex reduction. Peroxide is responsible for immunoassay marker enzyme. And beta amylase is a maltose syrup preparation. It is helpful in the preparation of maltose syrup. Okay, so A is the correct option here. Coming to the next section, that is the section B. In a large wild flower population, assume that no new mutation occur and no natural selection operates. What factors will affect the frequency of genotype in the population? Okay, non-random mating, gene flow, outbreeding within the population, invasion of new pathogen that kills a large number of individuals in the population. So they are asking no natural selection is operating here. But we see these all are the assumptions of natural selection. Okay, but they are asking no natural selection. Okay, so these all the options are correct because these are all the assumptions of natural selection. They're asking no natural selection operates. What factor will affect the frequency of genotype in this population? Okay, so these are all the correct options. Okay, which is or are the differences between microtubules and microfilaments? Microtubules are made up of tubulin and microfilaments are made up of intermediate filaments. No, this is wrong. Microtubules are made up of tubulin. This is correct. But microfilaments are not made up of intermediate filaments. Okay? Yes. Microtubules are important for compression resistance and microfilaments bear tension. So this is the correct option. Microtubules are important for functions of cilia and flagella and the microfilaments are important for cytoplasmic streaming. So this is also correct. Microtubules, muscle contraction, microfibrous ciliary movement. This is wrong. Microtubules is for ciliary motion and microfilaments is for muscle contraction. Okay, so this is the wrong option. So B and C are the correct options. N and N naught represent the number of viable cells at time t during sterilization and the start of sterilization t is equals to 0 respectively. Assuming that the cell that follows the first order kinetics and k is the death rate constant, which one of the following relationships is correct? So here this is the correct option. This is the equation we know, integration of minus integration n by n naught is equals to k into t. So this is the correct option. 19th question. Which one of the following statements about antigen antibody complex are true? Hydrogen bonds and van der Waals force participate in the antigen antibody interaction. So this is true. In the antigen and antibody interaction, it has to be purely non-covalent bond. Okay, so these both are the non-covalent bonds. So this is the correct option. Okay. Ionic bonds and hydrophobic bonds participate in the antigen antibody interaction. Okay. Yes, these both are also non-covalent bonds. So this is also correct. The combined strength of all interaction between single antigen and binding site on an antibody and a single epitope is called avidity. No. See, till here they have given the correct. But a single epitope is called avidity. No. All the epitopes, the strength for all the epitopes is known as avidity. See, if this is an antibody, this is binding to one antigen. So this is called affinity of this antigen antibody. This is calculated for only one antigen and one antibody binding site, single interaction. But avidity is for the whole antibody. Suppose this is a like this antibody is like this dimer. Okay, so now this is calculated for this whole, how much affinity total it is having, for this. This is known as avidity, but here they are telling single epitope is called avidity. So this is wrong. Antibody elicited by one antigen can cross react with the unrelated antigen. Yes, this can happen. This can happen when the antibody produced by one antigen is having more affinity for the another antigen, then this can have a cross reactivity with the another antigen. When it is having more, affinity for that antigen. Okay, so that's all for today. Thank you.